Okay, I'm going to lump a few slides together because they all kind of fit into the signal transduction, whether or not it's ligand binding or whether it's lipid rafts, which is a hot new topic. But the next three slides all relate back to the general overview of signal transduction. So, uh, what you need to think about is that the ligand binding will cause conformational changes in the receptor proteins. And those conformational changes will allow other things to occur, whether it's an activity on the receptor or whether it's an activity by the receptor. It requires the binding and conformational change that's induced by a ligand binding to receptor sites on the outside cytoplasmic side of the cell. So they can cause clustering of receptors. So it causes the receptors to get a little bit closer together because of conformational change, or it can increase phosphorylation or dephosphorylation, and it can cause kinase activity to be stimulated because of the conformational changes. And that will induce some sort of response inside the cell that's usually a signal transduction mechanism. Now, both B and T cell receptors. So, B cell receptors are basically future antibodies, is what we think about, but they have co-receptors that are associated with them, and T cell receptors do the same. If you notice, the carboxyl tails that are coming on the inside of the cytoplasmic portion of the cells are too small, too short to actually induce any kind of receptor mechanism from occurring. So these other cofactors will allow you to change up the cofactors that are interacting with the primary receptor, like the antibody or the T cell receptor. And by changing those up, we can have several different responses from the cell based on how tight of a binding we've got to a ligand on the outside of the cell. Um, so what happens is the antigen binds. In this case, it's expressed by the MHC2 over here, and then you've got the TCR. And that binds, but it requires some sort of cofactor, like these CD3 cofactors that can bind to the surface of this cell in order to stimulate them to send a signal transduction into the cell. Same thing with the ITAMs that are shown on these co-receptors on the B cell. It requires this binding and co-binding reaction to occur in order to send a signal into the cell. Now, this is one that I really think is a hot, and I call it a new topic, but it's not really that new. It's been known for a decade or so. We used to think that the fluid mosaic model of cellular protein organization was just random floating around of these proteins in the surface of the membrane. Well, that doesn't really make sense, because if you're putting all of the things you need together to make a cake, you're not going to scatter them all over the surface randomly of the kitchen. You're going to want them to be in one area so that you can cook that cake as efficiently as possible. So what they started studying and finding out is that the types of lipids that are found in the membrane can vary, and you can have a raft of very specific types of lipids in which these cellular components, whether it's a B cell receptor, primitive antibody, and its cofactors, will associate in that raft. So now you've moved them from being random to binding and activating all together in a small space on the surface of the cell. That's kind of cool. So without the antigen being bound, they may not be associating within the raft. But once they are bound to the antigen, they're going to be more attracted because of the conformational changes that occur. And that allows the signal transduction to be internalized, and you get a signal response. That's great because it prevents oversensitization of these chemicals when there's no antigen present. It's just another regulatory mechanism that's found within the cell. So be sure that you can verbalize what a lipid raft is for your exam. I don't know if I want to talk about this too much. You do have SRC or SARC kinases, um, and they're associated with avian cancer. The poultry industry is really interested in these right now. So SARC's involved in cell proliferation or cell signaling. We really don't know for sure. I think it's more cell signaling that induces cell proliferation. You don't need to know that particular one. If you were to take a membrane biochemistry course, you'll have to learn the structure of the phosphatidylinositolamines, um, whether they're biphosphate or triphosphates, and then the phospholipase C acts on that and it produces diacylglycerols. But I'm not going to make you memorize that for